recording? Okay, I'm recording now. It is the 1:45 the Wednesday, September 18th. And then we are gonna go to finish share the remaining of chapter 5. Okay, so your quiz on next week, even though I'm gonna go ahead and start on on chapter I'm gonna start on chapter 9 sometime next week, but this is chapter 5. So we did talk about I need to talk about the membrane, all of the details that what membrane of a cell or organelles. We talked about these things. Membrane structure is phospholipids arranged in a bilayer, includes glycerol, phosphate, and sphingolipid. Globular proteins inserted into the bilayer and fluid mosaic model. They are forgetting one other thing, which is cholesterol. Also, cholesterol is in the cell membrane. So this is pretty much the structure of cell membrane. There are protein molecules outside which are called extracellular matrix, ECM, right here. And there are protein molecules inside of the cell membrane, which are referred to as cytoskeleton, what holds up the cell. And there are protein molecules embedded like a chocolate chip cookies right here. These are the chip inside of an Oreo cookie. Imagine the Oreo cookie is bilayer, is 2-layer, the chocolate outside, and the white inside. Okay, and now embed, stick some chip, chocolate chip into Oreo cookies. That's what the structure of cell membrane is like. Okay, so anyhow, we talked about all of this. And then phospholipid bioavailable flexible matrix, barrier to permeability, transmembrane proteins, integral membrane proteins, interior proteins network, peripheral or intracellular membrane proteins, which we'll talk about all of these. Cell surface markers, glycoproteins and glycolipids, which we talked about that a little bit. The in your red blood cells, your blood type A, blood type B, blood type AB, blood type O. He's talking about these cell surface markers. So, and here's a transmission electron micrograph. Here's a phospholipid bilayer. Here's a phospholipid bilayer. So, and this is the one cell. This is another cell. Cytoplasm of one cell, cytoplasm of another cell, cell membrane, and the green in between is the interstitial, we call it interstitial fluid. Here they are showing you all of the structures. I talked about that. Lipidemic, it means defines the number and biological functions of lipids. 1,000 distinct lipids in cell are divided into 3 classes. Glycerol phospholipid, sphingolipid, and the sterols. So here it is that sphingolipid. We talked about that last time. I thought it was phospholipid, but it is structural wise is a little same thing. In general, is same thing. Hydrophilic, it loves water, and then hydrophobic tail, same thing. Phospholipid spontaneously formed by a layer because of the amphipathic. It means it has the head, we talked about this last time. The head loves water, the tail does not like water. Here's a phospholipid. This is where we stopped. Okay, so the phospholipid bilayer are fluid. It means they do move. It's not static. The wall behind me are static. They do not move. But the human being who lives in this house are fluid. They come, go, move around. I hope I defined the term fluid and static for you very well. Static does not move. Fluid moves. So hydrogen bonding of water to itself and to polar heads holds the layers together. Okay, so here they've done scientists have done an experiment. This is human cell and this is a mouse cell, and they tried to fuse them together. They saw, yeah, they can fuse together. So that is saying individual phospholipids and unanchored proteins can move through the membrane, and that experiment proved it. So protein molecules in the phospholipid are moving. The phospholipid itself, they're moving. Okay. Influence of fluidity of the phospholipid bilayer. Saturated fatty acid make the membrane less fluid than unsaturated fatty acid. So saturated fatty acid, remember, they don't have the kink. Unsaturated fatty acid have a kink. So when you have molecules are next to each other like this, it is hard to penetrate through. But when you have a molecule like that, it is easier to penetrate through. And that's what that sentence is saying. Saturated fatty acids make the membrane less fluid than unsaturated fatty acid. Okay, less fluid, less movable. Okay, warm temperature makes the membrane more fluid than cold temperature. Cold tolerance in bacteria due to fatty acid deacetorases. That's an enzyme. And yeah, I'm not familiar with what is fatty acid deacetorases. I'm not sure what that enzyme is. Anyhow, lipid composition of the ER and the plasmic reticulum and Golgi stack, Golgi apparatus, and plasma membrane are distinct. Composition affects fluidity, thickness, shape of the membrane. Okay, here they are in the plasmic reticulum and the PM, plasma membrane. The differences he's talking about, the differences major packing defects thin, but they have more cholesterol here, saturated fatty acid, more of it here, more of unsaturated fatty acid. Okay, and the protein molecules usually 25 amino acids. Here is 20 amino acids. Okay, so inside of the membrane, anionic head, it means they're negatively charged, but in the endoplasmic reticulum, you do not have that. You have a few outside. But here, toward the cytoplasm of the cell, there are more negatively charged molecules. But anyhow, not a big deal. I don't ask you much about what are the differences between endoplasmic reticulum and plasma membrane, cell membrane. Membrane proteins, their functions, very, very important. I will ask you a lot about that. We need to know this for rest of semester. Transporters, they are enzymes, cell surface receptors, cell surface identity markers, cell-to-cell adhesions, proteins, attachment to cytoskeletons, and affect membrane structure. So throughout the semester, we will talk about this, about most of these things. Well, mostly this chapter and chapter 9. And then throughout the semester, you will see it here and there a little bit. Okay, here is a transporter. They act as a gate. They open up and close. So molecules can go from outside of the cell to the inside of the cell. They act as an enzyme. These two molecules, it converts the triangular molecule to a circular molecule. Cell surface receptors, some of them like HIV, The white blood cells have the receptors for HIV, the virus HIV you're familiar with. But the liver cells do not have it, the receptor. The cells in your lungs have the receptor for COVID virus. The cells in your brain do not. For example, okay? Cell surface identity. Here are the carbohydrates. You remember hexagon carbohydrate attached to protein molecule. Example of that would be red blood cells that are blood type A, blood type B, blood type AB, blood type O. Okay, you will see that. Cell-to-cell adhesions, when one cell is attaching to another cell, right here. We studied 3 of those before. It was the gap junctions, desmosomes, and then attachment to cytoskeletons. Some of the proteins are attached to cytoskeletons. We did talk about those two. Structures relates to functions. Diverse functions arise from the diverse structures of membrane protein molecules. Yes, so the liver cells are different than your white blood cells because of their structures as well. Have common structural features related to their role as a membrane protein molecule. And they have a common structures, 2 layers of phospholipid, but the protein molecules makes a difference. Okay, anchoring molecules. Anchoring molecules attach membrane proteins to membrane surface. Anchoring molecules are modified lipids and non-polar regions that insert into internal proteins of the lipid bilayer, and chemical bonding domains that link directly to proteins. So what I mean by anchoring molecules, you've seen it right here. Oh, come on. Right here. These are anchoring molecules that they anchor to cytoskeleton. But other molecules, they are not anchored. They can freely move around. That's what he's talking about. Okay, transmembrane proteins, they go through phospholipid bilayer. This is a transmembrane. This is phospholipid bilayer. So it goes through both layers. So nonpolar regions of the protein molecule. So this would be the nonpolar region. This would be polar region. This would be nonpolar region of the protein molecule. Remember, this is phospholipid bilayer. So this end is inside of the cell. This end is hydrophilic. This end, my nail, is hydrophilic. But right here, it's hydrophobic, the protein molecule. So the regions, the proteins are embedded in the interior of the bilayer, alpha helix, beta sheeted pleated sheets, and polar regions of the proteins protrude from both sides of the bilayer. Yes, right here, this is hydrophilic, hydrophobic of the protein molecules. That's how it is, hydrophilic, hydrophobic. So transmembrane domain is a membrane spanning regions, hydrophobic amino acids arranged in the alpha helix. Hydrophobic, it's alpha helix right here. Proteins need only a single transmembrane domain to be anchored to the membrane. Yes, often have more than one such domains. Classifications of some types of receptors is based on the membrane. Yeah, do not worry about too much of this. Just the outside layer is hydrophilic inside, just like the phospholipid bilayer. Remember, this is the head of the phospholipid. Hydrophilic, hydrophobic, same as these protein molecules. Okay. Pores, it means there are openings. There are openings in the cell membrane. Extensive nonpolar regions within a transmembrane proteins can create a pore through the membrane. Cylinder of beta sheets in the protein secondary structure called a beta barrel, and interior is a polar and allows water and small polar molecules to pass through the membrane, right? Oh, it's not passive transports. It means molecules that are going inside of the membrane through these protein molecules that they do not need ATP, do not need energy. That is called passive transports. No energy needed, no ATP needed. Molecules move in response to a concentration gradient. Yeah, because when this happens, where more of a molecule are, they move through a membrane, move to where less of that molecule is. It is just like the lab you've done with dialysis bag. Okay, we didn't do anything with the Elodia. Brought it in the lab today, but you guys did not have time to do it, but I will bring it again on next Wednesday. I would like you to do it. Add some salt solution, look at the Elodia leaf and see what happens. Okay, so diffusion is movement of molecules from high concentration to low concentration, will continue until the concentration is same in all regions. Right here, we did this in the lab. You guys remember this? Diffusion. And then transport across a membrane. There is no membrane here. Right? But let's say there is a membrane. What will happen? Major barrier to crossing a biological membrane is the hydrophobic interior that repels polar molecules, but not nonpolar molecules. Nonpolar molecules will move until the concentration is equal on both sides. Limited permeability to small polar molecules. Yeah, very limited permeability to larger molecules. Large molecules cannot move through the phospholipid bilayer. Polar and small molecules can move through. There are pores in the membrane. Okay. Nonpolar molecules will move until the concentration is equal. I said polar, I'm sorry about that. Nonpolar molecules. Okay. Facilitated diffusion, it means that the protein molecules are involved and the molecules go through the protein molecules without any ATP. Molecules that cannot cross membrane easily, they move through proteins molecules. Move from high concentration to low concentration. They call them channel proteins like water. Hydrophilic channels, when open, carrier proteins bind specifically to molecules. They assist like glucose or sodium, and they move across a membrane. Membrane is selectively permeable because of channels and carriers. That's right. I had a physiology professor in Kentucky. He used to say, cell membrane has brain. I said, what? He said, yeah, they're selective. They can choose what molecule go through and what molecule does not go through. They do not have membrane, but they do not have brain. But and then finally we have ion channels. The protein, those channels are protein molecules within the phospholipid bilayer. Allow the passage of ions through nonpolar interior of the plasma membrane. Gated channels open or close in a response to stimulus, chemicals or electrical. 3 conditions determine directions: relative concentration of either side of the membrane, voltage difference across membrane, and gated channel. Is channel open or closed? right here. Facilitated diffusion. I like pictures more. Here is a gate. It opens and the molecules go through from high concentration to low concentration. Okay, or from inside of the cell, they go outside without any ATP needed. Remember, you guys, this is not diffusion because because a membrane involved. This is not diffusion. This is, they call it facilitated diffusion. It means through a membrane and no ATP needed. Okay, other pictures. Oh, carrier proteins. They can carry molecules from one end of the membrane to the other end, from inside to outside or from outside to inside, right here. Here's the molecules outside of the cell, and the carrier molecules will no ATP needed. Facilitated diffusion by no ATP needed. So the molecules go through and the gate opens and closes. It changes its shape. Still, it's not an enzyme. It changes its shape and the molecules go inside of the cell. Okay, osmosis, we talked about, we did this. Cytoplasm of a cell is an aqueous solution. Water is solvent. Dissolved substances are solute. Osmosis is net diffusion of water across a membrane toward a higher solute concentration. That's the definition I gave you. I gave you the definition of osmosis. I gave you movement of water molecules from high concentration to low concentration through a membrane, wherever water molecules are more, from outside of the cell to the inside of the cell, or from inside of the cell to the outside of the cell. There is osmosis through a membrane. Here is they're showing you a diagram of there is more solute on this side of the tube than this side. So what will happen, water molecules go from this side, from high concentration to low concentration, because there is more solute here. So osmotic concentration, when two solutions have different osmotic concentration, hypertonic solution have higher solute concentration, hypotonic solution have low solute concentration. And when two solutions have the same osmotic concentration, which was your bag B, I believe, in the lab, refer to your lab report that you wrote. Look at it. Aquaporins, specialized channels for water in cell membrane, is called facilitated osmosis. So those protein channels in the cell is called aquaporins. Important term to know. Everything in this page is important to understand and to know. Osmotic pressure, forces need to stop osmotic flow. Cell in a hypotonic solution gains water, causing cell to swell, creates pressure. Cell wall can reach balance of osmotic pressure, driving water in with hydrostatic pressure driving. I like to see a picture to explain osmotic pressure. So prokaryotic fungi, plants, and many protists, they do use that osmotic pressure. Plasma membrane is not strong and may burst, so animal cell must be in isotonic environment, but they are not. They have a system to, in case of protista, they have a system to get rid of the excess water, which I did talk about it. Okay, here they're showing you a few scenarios. For example, you put human red blood cells in a hypertonic, in a salt solution. What would happen? The cells shrink. You put human in a normal solution, same as your body. Nothing happens to the cell. You put human in a hypotonic solution, the cell becomes enlarged, burst, and die. Okay, same thing as plants. The plant cell will shrink. Here is the plant cell in isotonic environment. They will flaccid, they shrink if they are placed in isotonic environment, and in a hypotonic environment, because they have to transport water. They are normal target cells. They stay normal. That's what happens. In the story in plants, it's a little bit different, and I'm not too concerned. I guess when you take botany, they will talk about it a little bit more, but I would like you to know these three things. Shriveled, normal, and cell will swell. Maintaining osmotic balance. Of course, plant cells, I talked about this a little bit. Active transports. Yeah, I'm not too crazy about maintaining osmotic balance. It's okay. It's good information, but I'm not too worried about it. Active transport, very, very important. Active transport, you remember so far whatever I talked about? Molecules move, do not need ATP. But now I'm talking about movement of molecules that across a membrane that needs ATP. I hope I'm making sense in that regard. So carrier proteins and active transports. Carrier proteins used in active transport include uniporters, symporters, and antiporters. Move one molecule at a time, move two molecules in the same direction, and move two molecules in opposite directions. Okay, I hope your textbook just did not wrote them down. They have some pictures. If they have pictures, then I would like you to know that. If there is no pictures, there is no diagrams, forget it. Okay, terms can be also used to describe facilitated diffusion carriers. Okay, so yeah, okay. So sodium-potassium pump is an example of antiporters. Molecules are moving opposite of each other. Okay, so direct use of ATP for active transports uses antiporter to move 3 sodium outside of the cell and 2 potassium inside of the cell. Movement is against the concentration gradient. ATP used to change the conformation of the carrier proteins, affinity of the carrier proteins for either sodium and potassium changes, so ions can be. When you take physiology, they talk about this a little bit more. Here, all I have to say, just know this, because then I guess when I talk about neurons, how neurons are, sodium is outside of a neuron and inside of a neuron is negatively charged, then this comes to a picture and it will explain things a little bit better. But for now, just know sodium-potassium pump move things in and outside of the cell right here. Sodium-potassium pump, ATP is needed, and they're showing sodium by square. So sodium is attached to here, 1, 2, 3, and move out, and potassium are moved in at the expense of 1 ATP. Okay, coupled transports use ATP, uses energy released when a molecule moves by diffusion to supply energy to active transports of a different symporter or antiporter can be used. Glucose and sodium. Symporter captures the energy from sodium, diffuses to move glucose against its right here. Yeah, okay. So he's talking about symporter as well too. So here the sodium potassium pump, here is the glucose and sodium. So sodium is moving inside of the cell, and glucose is moving inside of the cell at the same time as sodium is moving inside of the cell. Again, this is a different protein molecule than this one. This different cells have this one, and different cells have that one. Possibly some cells might have both of them. Okay, so it is a symporter. And then quiz question or exam question: does symporter need ATP? Answer is no. Right here, ATP is needed here for porter, but for symporter, no ATP is needed. Okay, bulk transport. Okay, your textbook did not give you an example of, there were 3 of them. You remember uniporter. Uniporter water molecules is an example of aquaporin. I gave it to you. I said, make sure you know this one. It was this PowerPoint's osmotic pressure. Anyhow, there was one, aquaporin is an example of uniporter. So yes, your textbook gave, they do not have a diagram of it, but they gave you example of symporter and antiporter. So I would like you to know those. Coupled transport, we talked about this. The big bulk, yes, it is important. Endocytosis and exocytosis. Endocytosis when the molecules move inside of the cell. Exocytosis when the molecules move outside of the cell. Okay, movement of substances into the cell requires energy. Phagocytosis, cell eating. Pinocytosis, cell drinking. Receptor, there are 3 types of endocytosis: phagocytosis, pinocytosis, and the last one, receptor-mediated endocytosis. Specific material can get inside of the cell. Specific molecules are taken in after they bind to the receptors. Exocytosis, when molecules get outside of the cell. Like you release hormones from your cells. They go out through exocytosis from your pancreatic cells in your pancreas. Insulin get out. That's exocytosis. It requires energy. Movement of substances outside of the cell. Here's a bacteria being engulfed. A cell membrane pinches, and a cell membrane forms vesicles around the bacteria. That is phagocytosis. Here's a picture of it. Phagocytosis, you have little solutes, very small molecules. The same thing, same process again. Form vesicles around it and goes inside of the cell. Phagocytosis, pinocytosis. Receptors mediated, it means these are target molecules. They sit on these receptors. This is a cell membrane. And these are protein molecules. The receptors are protein molecules. These target molecules sit on the receptors, and then a coat is formed. And that coat goes inside of the cell. Okay. I hope I'm making the differences. Exocytosis, a vesicle is formed. This inside of it, neurotransmitters, hormones, what have you, and they come and they diffuse, they attach to the cell membrane, and now this vesicle, the membrane of it's phospholipid, attaches the cell membrane. So cell membrane constantly being remodeled by these vesicles. Okay? And that should end the chapter. So we will talk about chapter 5, 6. Sorry about that. No, I'm done. No, not chapter 6. I'm sorry, chapter 9. Chapter 9, that's the next thing we are going to talk about. So this was chapter 5. I went over it. We need to go over chapter 9 because it is mainly about membrane. Chapter 9 is mainly about membrane. All right. I'll see you later. You have a good day. And I'm going to stop recording.